Okay, so welcome back to the second part of the videos on exchange surfaces. Now, if you haven't seen the first part, then please do go back and watch that, because I go through exactly what makes a good exchange surface and, of course, what an exchange surface is. We also looked at a couple of examples, and in this video, I'm just going to have a look at two more. So let's crack straight on. And the first thing we're going to look at is the leaf, right? So a leaf definitely has more than one exchange surface. Right, there are certain things that need to be exchanged by the leaf. Those include carbon dioxide, okay, because plants need carbon dioxide in order to photosynthesize. Oxygen, because plants produce oxygen in photosynthesis, right, and then they need to give it back out because it's a waste product of theirs. And water vapor, right, water vapor. Okay, because plants obviously lose water vapor via transpiration. That's basically where water is evaporated from the plant. And this needs to be managed, okay? Sometimes plants want to lose water, sometimes they want to retain water. And all those things obviously need to be exchanged. All right, now we're going to have a look at a more detailed diagram. Basically, if we were looking side on at a leaf, and you zoomed in and you were able to see the cells, that is what we're really going to look at, because the main exchange surface for a leaf is on the bottom of the leaf. Okay, let's scroll down. And here we go. This obviously looks kind of different, but this part here is the top of the leaf. This part here is the bottom of the leaf, right? This part at the bottom is going to be our first exchange surface. All right, so you'll notice over here, okay, this here, I'm going to rub these arrows out because that was just for demonstration. These here, okay, let me just circle it like that. Those things, they are called guard cells, right? Those are guard cells. They are cells that actually control tiny openings in the bottom of the leaf, right? So this here, you'll notice that there is a hole there. That hole is called the, well, plural are called the stomata. Right, one is called a stomatum, plural stomata. Okay, so stomata. Now the stomata are extremely important because they allow things to come into the leaf and they allow things to be released out of the leaf. Okay, if we're thinking about carbon dioxide, well, carbon dioxide in the air wants to travel in. So if we've got CO2 in the air, that wants to come in, right? And it can fill up this air gap. These cells around that gap, they need CO2, right, in order to carry out photosynthesis, because the leaf is where photosynthesis mainly occurs. And so CO2 comes in by diffusion, okay, because there is obviously a concentration gradient from the air to the inside of the leaf. And then CO2, once it's in, will travel into these cells, right? And so that obviously is one example of exchange. Now, then what happens? Well, once photosynthesis occurs, etc., carbon dioxide no longer is there, okay, because they produce glucose, and the byproduct of that is oxygen, okay, so they produce oxygen, and so we have a buildup of oxygen inside the leaf, right, and inside the leaf, the concentration of oxygen gets to a point where it's higher than it is outside. And so the oxygen then diffuses out of the leaf. And so that is how this is acting as an exchange surface. Okay, something else which is also lost because it's also produced is water. Okay, you also get H2O, right? H2O is lost from the stomata. Okay, that is by diffusion. Now, I know that sounds strange because we said in previous videos that diffusion of water is basically called osmosis, but that's only true when it's across a partially permeable membrane. You'll remember we went through that definition. So if water vapor is traveling and it's not going through a partially permeable membrane, then we do call it diffusion. Okay, when water travels across a partially permeable membrane, then it's called osmosis, right? So there is a subtle difference there. Okay, so we've actually seen two exchange surfaces there. One is this, the bottom of the leaf, okay, where we're going to exchange gases in and out. And the other is the inside of the leaf, okay, so across here. And you'll notice that there is quite a high surface area because there are all these air gaps, right? All these air gaps mean that more of the cells are exposed. Okay, that means that more diffusion can happen in the same period of time. The diffusion rate increases because we've increased the surface area. Right, leaves themselves, if we scroll back up, you obviously know what a leaf looks like, but leaves themselves are this flattened shape, okay, rather than being really thick. they're really flat so that they increase their surface area to volume ratio. And you remember that that is one of the key things in order to increase the rate of diffusion. Okay, so they have a higher surface area to volume ratio, makes them a better exchange surface. Okay, and the last point I wanted to make is these things here, I mentioned at the start, the guard cells. Those guard cells basically have the ability to either open or close these stomata. This hole is not always open. Okay, what they can do is actually push together. I'll just draw it in green. They can push together and fill in that gap. And now it's much more difficult for things to get in and out of the leaf. Okay, this happens most often when the leaf is losing a lot of water. So let's say, for example, it's really dry outside and it's really hot. Water is actually going to be lost really quickly. Yeah, remember we said temperature actually increases rate of diffusion as well. The leaf is going to dehydrate quicker. As a reaction, the guard cells close the stomata and therefore reduce the rate at which water can leave the leaf. This is obviously important because if this didn't happen, the plant would dehydrate and it would wilt. Okay, what that means is basically dehydrate and it like shrivels up. Okay, but the plant would wilt. If you want the spelling for that, it's W-I-L-T. Okay, so the guard cells actually stop that from happening, and so that is very important as well. Alright, so let's have a look at our last example of an exchange surface. And this here is meant to be a fish, right? So this is basically, and these here are the gill filaments of the fish, right? So the way that fish obtain oxygen into their blood, they don't have lungs like we do, they have gills. So what happens is water, let's just draw it in here, water comes through the mouth like so, okay? And then it goes out and it goes through past these gill filaments, right? We call them gill filaments. And then they escape out of the fish via the gills, right? So that's why fish have gills. It's actually water leaving via the gills and entering via the mouth. Okay, but what actually happens, or your actual exchange surface, are these things here, okay, the gill filaments. So we'll have a look at one now. Alright, so let's have a look. So this is a very simple diagram of a gill filament, right? This in 3D, okay, actually, let's draw it in black, like so. In 3D, this gill arch here, basically it's a stack containing more than one of these things, right? I'm not really an artist, so I can't draw it, but imagine in 3D, you've got another one coming down, etc. And they're all stacked up like that. Okay, so there's not just one of them, there are quite a few. Now, these dark red things here, okay, there and there, are called the lamellae, right? Singular lamella, plural lamellae, right? These are called the lamellae. Basically, that is where the gas exchange is going to happen. You've got a good blood supply, so a capillary network in those lamellae, right? And basically what's going to happen is the water is going to flow over them. Now, the reason for this, by the way, is that the water contains oxygen. There is oxygen dissolved in the water, okay? And you have a bed of capillaries in here, and the blood runs in the opposite direction to the water, and this is very important, right? So the water, you can see the arrow there, but let's just put some more on. The water, let's say, is going from bottom to the top there. Now, if I were to draw a capillary, right, obviously not to scale, like that, what will be happening is the blood will be flowing this way, right? And with the blood flowing in the opposite direction to the water, what that does is it maintains the concentration gradient, okay? Because it means as the water's going through, it's reaching new blood which does not have oxygen, and so more and more oxygen can just diffuse into the blood as they're rushing past each other. If they were going the same way, then obviously diffusion would occur until the concentrations were equal, and then diffusion would stop. But this way, it means that all the oxygen from the water can diffuse into the blood, and that makes it much more efficient, right? So remember, in the last video, we said that maintaining a concentration gradient is really important. This is an example of a system which is adapted to maintain the concentration gradient. Right, the fact that these are really flat and they're all stacked on top of each other also increases the surface area. That means we have a large surface area in order for diffusion to occur, and that obviously increases the efficiency of the surface as well. Okay, so I'm going to stop there. I only wanted to go through those two examples and make sure that you were clear on them. But if anything is still unclear, please do feel free to post a comment in the box below or send me a direct email using the link. But otherwise, please do like and subscribe because it really does help me out. And of course, you'll be notified when new videos become available. But I look forward to seeing you in the next one.